flowing on from comparative DNA and comparative genomics, it's important that we scale this up and we have a look at now comparative proteins and also this idea of bioinformatics. So comparative proteins, what are they? So we already know that proteins are long chains of amino acids bound together by a peptide bond. And what we're really looking at with comparative proteins, very simply, is just looking at how that sequence of amino acids in a protein is determined by the sequence of bases in the DNA. So we're linking it to that prior lesson. Now, in the prior lesson, we spoke about having a script ready to go for short answer and extended response questions when it comes to answering evidence of evolution and how these different examples can be used as evidence of evolution. So when we're having a look, fewer differences, again, this time, even while we're looking at comparative proteins, in the DNA sequence or in the amino acid sequence or in the sequence of, or in the protein itself, indicates a closer evolutionary relationship between species, a more recent common ancestor, and less mutations have been accumulated. Now, this here is a very quick demonstration from the textbook just showing you the different three-letter codes that we use for each of the amino acids. You do not need to know the amino acids, so that's really important. Do not waste your time with them. So what we have here is something known as a ubiquitous protein. So this is a protein that we can use in comparative proteins as they appear in all species. Now, generally speaking, they perform really, really simple functions, okay? And the function tends to be the same no matter where they're found. So it could be found in a yeast molecule, a yeast cell, it could be found in a human cell. Ultimately, this ubiquitous protein would perform the same job. So one particular example of a ubiquitous protein is known as cytochrome C. So cytochrome C is a ubiquitous protein involved in cell respiration, and in humans it contains 104 amino acids. So what we can do as evidence of evolution is compare how many of these same amino acids are found in the same position in cytochrome C over different organisms. So in the table on the right here, we can see that if we have a look, the chimpanzee, the gorilla have no differences with us. They contain the same 104 amino acids in the same sequence as humans, indicating minimal difference, or literally no difference, right? And so this suggests to us that they must have come from a more recent common ancestor. As we look towards the bottom of the table and we look at, say, yeast here, yeast has 44 differences compared to human cytochrome C. So it indicates that comparing humans to yeast indicates a very distant common ancestor and not nearly as related as a chimpanzee or a gorilla to Homo sapiens. So in the second part of this lesson, we're now going to look at bioinformatics. The definition to this is important. It has been known to come up from time to time, and it's important you know it, and you can quote what I've put in bold here, the specific fields that bioinformatics includes. So what is bioinformatics? It's not a single field, it is a multidisciplinary field combining biological science with, number 1, computer science, 2, engineering, 3, statistics, and 4, mathematics. Please learn this. Please go through, take the time to understand what this is. Now, it's all well and good you understanding what it is and being able to quote a definition for what bioinformatics is, but you also need to be able to explain it, okay, and link it into the scenarios that we use. So why do we use this? Why are we linking biology with these 4 main areas? Well, the most essential part today is if we're analyzing entire genomes, we are going to get large amounts of data. And so we need to use computers, we need to use clever statistics programs, math, and even some forms of engineering to really help us with managing the data that we get from sequencing whole genomes. If we individually went through and we counted, okay, humans have roughly 3 billion base pairs, it would take us almost forever, okay? But with computers, we can save the amount of time and we can get a much better understanding of what those 3 billion base pairs do when we can sequence them much